The cell cycle is made up of two parts that constitute the entire life cycle of a cell. The first part is interphase, which is the period of time between cell divisions. It's going to be the longest phase of our actually 3 phases of the life cycle of our cell, and it's going to include steps for growth and basically doing the job inside the cell. Inside the organism, or the job of the cell if it's a single-celled organism. It's going to include DNA synthesis, which is chromosome replication in preparation for division. And then we've got M phase, which is going to be our mitosis or meiosis when we get there, which is going to be division of the nucleus, and then cytokinesis, which is technically its own separate phase from mitosis. It's not part of mitosis, and it is the separation or the splitting of the cytoplasm. The cell's life cycle starts at the very end of the previous division. So basically you can kind of think about that as like the cell birth, and that's going to be the start of interphase. Interphase is made up of 3 subphases, 2 gap phases, and 1 synthesis phase. The first phase is going to be the gap 1 or G1 phase. In this phase, this is where most of the growth of the cell is going to take place. It's where all of the gene expression is going to happen. The cell is doing its job, whether that is part of an organism or as a single-celled organism. And so it's expressing all of the proteins that it needs. It's expressing all of its RNA for expressing proteins or for gene regulation. It's performing all of its metabolic activities. We'll talk about checkpoints in just a minute. Checkpoints are basically time in the cell life cycle where it's a check-in to make sure that the cell is healthy and it's ready for the next phase. I'll talk about those on a diagram coming up. After G1, the cell enters S phase. S phase is where DNA synthesis or DNA replication takes place. We're not going to learn about replication right now. We're just going to keep in mind that it happens. And the key thing here is that we are making enough DNA so that we can partition it into all of the cells that we're going to produce at the end of either mitosis or meiosis. When our chromosomes are copied, they are going to be copied and joined with an identical copy called a sister chromatid. We'll come back to that in just a minute, but generally, following the S phase, the cell enters the gap 2 or G2 phase. The main thing that happens here is preparation for cell division. So synthesis happens in preparation for cell division. And then once the synthesis is complete, the cell says, cool, I'm ready to go. Let's start building all of the things that we need for cell division to actually take place. The cell may still be doing some of its job. It's probably still doing some gene expression, but it's doing a lot of really ramping up, making all of the tools and the enzymes and all of that it needs to start this process of mitosis. Again, I will talk about checkpoints in just a minute. If you want to go ahead and write down what this checkpoint does, we'll come right back. Here we have a diagram of our cell cycle. We've got a cell here that has just about to finish going through mitosis. It's going to finish mitosis, and we've got 2 cells at the end of mitosis. And we're going to follow one of them through its life cycle all the way through to the end of mitosis. So keep in mind the other cell is going to go on, enter G1, and then start doing its own preparations for cell division. Mitosis is only a small portion of the entire life cycle of the cell. If you think about the average cell in the human body, like let's say a skin cell, skin cells replicate roughly every 24 hours. Mitosis takes about 1 hour of that 24 hours in skin cells. All cells are going to be a little bit different, but they all kind of have mitosis as the smallest portion of their life cycle. So we'll start at the end of mitosis. Our cell is going to go through G1, where again, it's doing its metabolic work, it's expressing its genes, it's living its best life. Once it starts to approach S phase, it's going to enter the G1 checkpoint. A checkpoint is basically a gate. And the cell is going to take stock at this point and just kind of see if it is ready to go. And if you go back a couple of slides where I mentioned the G1S checkpoint, there's a little description of what happens at that checkpoint if you want to get it down in your notes. But basically, the cell here is checking to make sure that it is ready for DNA synthesis. And it's also going to check to make sure that the cell is growing properly, that it's big enough, that it's healthy, and just kind of make sure that it's ready for this really energetically expensive process of DNA replication. So assuming that everything is good, the cell's going to cross that checkpoint and continue getting ready for synthesis, and then it is going to switch over to DNA replication. Again, we'll talk about replication later in the semester, but we are going to take one chromosome and we are going to copy it into what we call sister chromatids. And this is going to happen for every single chromosome. And now we have all of our pairs that are ready to be separated during mitosis. And these guys are going to stay together through the end of S phase and then all the way through G2 until we get to mitosis. Once S phase is complete, the cell is going to enter G2. It's going to start preparing for cell division, making all of the enzymes, making all of the components that it needs. When it's done with that, it's going to reach the G2M checkpoint. At this point, this is kind of the last checkpoint before the cell really starts going into mitosis. The cell is going to check to make sure that DNA has been properly replicated. And make sure that all of the components are present. So we're going to need a structure called a centriole. The centrioles are going to produce microtubules, which are going to move the chromosomes around. We need the enzymes in place to start breaking down our nuclear envelope. So all kinds of things have to happen in order for this process to move forward. So once the cell is good, take stock, make sure that everything is ready to go, then it can enter the mitotic phase. There is one more checkpoint that wasn't in your notes. This is in the middle of, or part of the way through mitosis, not quite the middle, and that is called the spindle assembly checkpoint. The spindle is going to be a structure called centrioles or centrosomes. I think our textbook used centrosomes. There is a distinction between the two. We're not going to worry about it right now. The centrioles, centrosomes are going to produce microtubules. And so we need those microtubules to attach onto those chromosomes and move those chromosomes around and eventually partition them. And so if that spindle is not ready to go, then there's no reason for replication to take place. If the spindle assembly is ready, then the cell's going to go through mitosis, finish up, cytokinesis is going to happen, and then we're going to enter back into interphase once everything is all done. If at any point the cell encounters a problem, it's going to stop at the checkpoint. And then one of two things can happen. One, the cell cycle can pause, until everything catches up. So maybe there was like a little nutrient deficiency for a while, and now we're good, and now we can get going. Or just something silly, something small, not silly, something small that just kind of trips up the cell. Great, the cell can pause, finish everything it needs for that G1S checkpoint or finish replication, whatever, and then it can continue. If it is a big deal, it can also stop the cell cycle, and then one of two things can happen. It can go into something called G0, or it can do something called apoptosis. And we're not really going to talk about apoptosis in genetics. That's more of a cell biology problem. Apoptosis is basically where the cell says, you know what, we're done, and then it's dead. So it can do that. Otherwise, it can enter sort of a long-term pause phase called G0. G0 is a non-dividing phase. And again, it can be permanent or it can be temporary. If it is a problem with the cell, it's probably going to enter apoptosis instead. But there are a few cells that will enter G0 on purpose. So one of those is neurons. So generally, neurons are produced and then no cell division. So they are produced by a division during development, and then they go through G1 for a while, and then they enter a G0 phase where they just sort of continue basically whatever was going on in G1, their growth, performing their job, expressing their genes. They just never prepare for mitosis. Another example of a cell that does this are liver cells. So liver cells don't divide unless the liver is damaged. And so once the liver is formed, it'll just enter G0 and stay there. And then if there's a reason to divide, then it will pop back into G1 and then continue. So G0 isn't really a default for most of our cells. Most of our cells are going to go through G1 and through their checkpoints and then pause or go through apoptosis if they don't continue. So during our S phase, we have DNA replication or DNA synthesis. I tend to call it replication, but it is synthesizing DNA. During this phase, we get the production of sister chromatids. So before we move on, I want you to just kind of recall from earlier in the lecture where we talked about homologous chromosomes. One of those chromosomes comes from the maternal parent or mom. The other one comes from the paternal parent or dad. So that's what M and P stand for, maternal, paternal. And they are chromosomes that derive from each parent. They have same size, same shape, same gene order, but they may differ in the alleles that they have. So let's say we'll just go big A's and little B's. So same genes, same place, same size, same shape, same centromere in the same location. During DNA synthesis, each one of those is going to replicate and remain connected at the centromere. So here we've got the maternal copy, identical copy, and the paternal identical copies. So if we were to draw this out, identical copies, we'll just represent that by different alleles there. And then they are going to remain together. When chromosomes are attached at their centromere, they count as one chromosome. So here on the left, we have 2 chromosomes counted by their centromere. On the right, we have 2 chromosomes counted by their centromere. This is by convention. It's to just keep down confusion because what we're really doing is doubling the genetic content, but they're always stuck together and they don't have a purpose in the cell. other than cell division. So these sister chromatids are basically a temporary situation, not extra genetic material. So we've got our chromosomes here ready to go. We've got our homologous chromosomes here on the right. I'm sorry, on the left, we've got homologous chromosomes. On the right, we've also got homologous chromosomes. They are just in their sister chromatid form. So we are all good for mitosis now. We've got all the way through gap 1, S phase, gap 2, all of our checkpoints, and now we are ready to enter mitosis, where we are going to divide the nucleus and separate those sister chromatids. And then we are going to, at the end of mitosis, enter cytokinesis, where we are going to separate our, or divide, split, however you want to say it, our cytoplasm. Our textbook doesn't really go into cytokinesis because our focus in genetics is the chromosomes and what's happening there. So you don't really need to worry about the mechanism of cytokinesis. For mitosis, I am going to go through each of the phases. I'm just going to go ahead and do it relatively quickly, and then whenever you are ready, just go ahead and pause and make your notes based on the images here. Mitosis cell division is to produce somatic cells. So somatic cells are our body cells. This is in comparison to our gametes, which we are going to produce during meiosis. So we're going to produce those somatic cells. And the goal here is to produce genetically identical cells. And we are going to maintain a diploid state. So we are going to start diploid and end diploid. This particular cell is diploid with 4 total chromosomes. And so we can count our chromosomes. We'll do it here in prophase. 1, 2, 3, 4, based on our centromeres. So starting at interphase, we've got our chromosomes. They are relaxed chromosomes because our cell has been using those chromosomes for gene expression and replication and doing its job. And so chromosomes are relaxed. Nuclear envelope is formed. We've got our centrosomes, which we're going to use in just a minute. Down on the bottom, we have an electron microscope, a colorized image that shows our nucleus in the center. And then these structures on the outside, those are stained microtubules. So microtubules function in several ways in the cell. One of them is movement, so moving proteins and vesicles and things around the cell, and then chromosomes during cell division, but also giving the cell structure. So that's actually what you're seeing here is the microtubules that are providing a 3D structure for the cell, and also providing basically tracks for things to move around in the cell. At the end of interphase, we are going to enter mitosis. The first phase of mitosis is called prophase. In this phase, our chromosomes start to condense. Remember, we're in our sister chromatid form, and our mitotic spindle is going to form from our centrosomes. So our centrosomes are going to produce microtubules specific for cell division. And they are located here at the top of this cell. Oops, that's the wrong color, sorry. Let's go with yellow. We've got them here at the top of our cell, and those are our little centrosomes that are growing microtubules out. You'll notice that they are starting to spread away from their point of origin to the poles on either side of the cell. As we move into our next phase, pro-metaphase, our chromosomes are going to continue to condense a little bit more. We've got those spindle fibers, mitotic spindle on either side, and we've got a couple of things going on with the microtubules. We've got microtubules that are growing and attaching to our chromosomes on either side, and they're coming from both sides. So we've got microtubules coming from both sides that are growing towards all of our chromosomes. And what they're going to do is tug of war those chromosomes as they move them. We've got another set of microtubules that are going to be growing all the way across. And what they're going to do is to help elongate our cell, because we need to make two cells, and so we need to spread out our cell, make it a little bit bigger so that our chromosomes can separate and we can push everything into two separate cells. So we're following our chart from the previous page. It went interphase, prophase, pro-metaphase, and then we came down from pro-metaphase into metaphase. So this one is going the other direction. During metaphase, the chromosomes line up along the center axis of our cell. That center axis is called the metaphase plate. And what really lines up is the centromeres. So our centromeres are going to line up along, and then the arms of the chromosomes are just kind of wherever they end up. You can see in the image down on the bottom that we've got our chromosomes lined up along the center here with the arms of the chromosomes just kind of spreading out. You can also see in this image the microtubules coming out of the spindle fibers. The mitotic spindle, microtubules coming out of the mitotic spindle and stretched towards those chromosomes. Once the chromosomes hit the metaphase plate, they are going to very quickly enter anaphase. Anaphase is our fastest phase. Once those chromosomes pull apart, they just whoosh to either side. They're pulled pretty abruptly by those microtubules. Basically, the microtubules act like someone reeling in a rope on a boat. When you're reeling them in and the the rope is just kind of piling in at the bottom. What's happening is that they're actually shortening, little pieces are coming off, but they are essentially just kind of reeling those chromosomes towards either side. When this happens, those chromosomes split. So very, very quickly, we go from 4 chromosomes in the center to 4 chromosomes that are each going to end up in their daughter cells. Now, we do not ever say that there are now 8 chromosomes here because anaphase is so fast, it's a blip. As soon as they pull apart, we enter telophase, and in telophase, everything sort of relaxes. Our nuclear envelope reforms, our chromosomes begin to relax, our mitotic spindle begins to break down, and cytokinesis starts to take place. So by the end of telophase and cytokinesis, we end up with 2 daughter cells. Each of those daughter cells are going to be genetically identical to each other and to the parent cell. And that is because our cells are trying to maintain the same cell type. So let's say we're a skin cell dividing into a skin cell. We want to make sure that we maintain all of the same genetic information that we had in that parent cell. So we don't actually want any differences here. So our newly formed cells have all of the chromosomes they need. We separated our sister chromatids, but we still maintain our homologous chromosomes. Which means that we also maintain that diploid state. The only difference from the parent cell is that our newly formed cell is a little bit smaller. It contains only half of the cytoplasm. And so the cell has to work to grow, produce more cytoplasm. That's going to happen in G1. We didn't talk about anything that happens in the cytoplasm. There are a bunch of organelles in there, and what happens to the organelles really depends on the organelle itself. A lot of organelle replication and sorting is, at least as of like a year or two ago, not very well understood. The last time I looked it up, there wasn't a lot of information on it. There hasn't been a lot of research in that topic. Anything that is sort of free floating in the cytoplasm is going to get partitioned. So mitochondria and chloroplasts, which divide on their own, get partitioned, and then they have to replicate in the new cells if they need more there. Most of our organelles are part of the endomembrane system, and so the membranes inside the cells have to be partitioned. And again, our textbook doesn't go into it. I've never actually seen a lot of information on it. We're not going to worry about it, but that is part of cytokinesis. This is a really great slide. It kind of shows you what happens during all of the different stages. It does address the number of molecules versus the number of chromosomes. So when they say number of molecules of DNA, they're talking about the number of chromatids. I also like that this has just a really nice diagram of what's going on. I feel like it's not quite as busy as our other mitosis diagram. Table 2.1 is a nice summary of what's happening in all of our phases in mitosis and in interphase. It's not necessarily all of the details, but it does give you the highlights. I did not mention that during metaphase, we actually meet that spindle assembly checkpoint. So that's where it happens because our cell needs to make sure that all of our chromosomes are attached to their microtubules and that the spindle has formed properly. So that is the end of meiosis. This slide was actually supposed to be in the next video, but it is what it is. Here it is. So we are about to move into sexual reproduction and meiosis. The goal of meiosis is to produce those haploid gametes. We need to produce gametes for reproduction, and those gametes need to be haploid rather than diploid because of fertilization. Because when the sperm and egg combine, they are going to combine their genomes. And so unless we want to double our genomes every time our sperm and egg come together, we need to start with less. So this happens for a couple of reasons. The big one is so that we don't double our genome. But the other thing that happens is that when we reduce our chromosome number, it also allows us to actually combine those genomes and introduce the genetic variation that comes from two genomes combining. And then on top of that, during meiosis, we are going to have a few points of genetic variation that happens during the process. And that genetic variation is one of the reasons why sexual reproduction is so successful and so many organisms use it. Asexual reproduction gives you a clone, basically, of the parent cell. Mitosis essentially gives you a clone. There shouldn't be any changes during mitosis, and generally there are not any changes during asexual reproduction. But during meiosis, we do have a few points where we get massive variation so that every sperm is different, every egg is different, which means every combination of sperm and eggs is going to be different.